In this topic, we're going to discuss enzymes and their inhibitors. So by the end of this topic, you'll understand what is an enzyme inhibitor, what is a reversible inhibitor, what is an irreversible inhibitor, and then you'll be able to describe how the rate of reaction is changed by each inhibitor. So what is an enzyme inhibitor? These are molecules which reduce the rate of an enzyme catalyzed reaction. How they do this is by interfering with the functioning of the active site of the enzyme, and they can either do this directly or indirectly. You have different types of inhibitors. You've got reversible inhibitors, which attach temporarily to the enzyme. You have two types. You've got competitive inhibitors, which bind to the active site of the enzyme, and then you've got non-competitive inhibitors, which bind to another position on the enzyme called an allosteric site. Irreversible inhibitors bind very strongly to the active site, so they can't be removed, and therefore they permanently prevent the enzyme from functioning. So an example would be heavy metals such as mercury and silver. So let's have a look at reversible competitive inhibition. Now what is this? Well, if you look here, you've got your enzyme, which is the big pink blob, and you've got your inhibitors, which are the little yellow faces, and then your little substrate, the blue face. So the inhibitor molecule, which is your yellow face, is structurally similar to the substrate molecule. So it can occupy the active site. So what does it do? It competes for the active site with the substrate, and for whatever fraction of time the competitive inhibitor molecule is occupying the active site, the enzyme is unavailable for catalysis. So what happens? Competitive inhibitors will decrease the rate of reaction. So an increase in substrate concentration, which you can see here with your little blue molecules, will increase the rate of reaction. The inhibitor, remember, is not permanently bound to the active site. And so when it leaves the active site, another molecule can take its place. This could be a substrate or inhibitor molecule, depending on how much of each is present. So sooner or later, all the substrate molecules will find the active site. So what are the different factors that affect the amount of enzyme inhibition? Well, in terms of competitive inhibition, the inhibitor concentration. So the greater the inhibitor concentration, what will happen? What do you think will happen? So if you increase the inhibitor concentration, the slower the rate of reaction. If you increase the substrate concentration, then there's more chance of the substrates competing with the active site. And then the last point is the relative affinities of the inhibitor and substrate for the active site. What does this mean? Well, it's the binding power of the substrate or the inhibitor for that active site. So here you can see another example of competitive inhibition. You've got your substrates, which are the orange molecules, and then your regulatory molecule, which is your inhibitor, represented by blue. So notice how when the blue regulatory molecule binds to the active site, the orange molecules cannot bind, and we say that they are competing for the active site. This is an example in your notes. The inhibitor is the pink molecule, and the enzyme is the blue molecule, your substrate is the green molecule. So notice how the shapes of the inhibitor and substrate are both very similar to the active site. So the inhibitor will compete with the substrate for that active site. And when it binds to the active site, this means that the substrate cannot bind, so it's competitive inhibition. Now we need to know an example. So an example we're going to look at is ethanol. In our bodies, ethanol is metabolized to something called acetaldehyde. Acetaldehyde is further oxidized to acetic acid, and the enzyme that does this is called aldehyde oxidase. Now this usually happens quite fast, so we don't get an accumulation of acetaldehyde in our bodies. So ethanol is broken down into or metabolized into acetaldehyde, which is oxidized to acetic acid. Now there is a drug called disulfiram that can help alcoholics. What it does is it binds to the enzyme and prevents acetaldehyde from being oxidized. This means that acetaldehyde builds up and it's got horrid side effects such as vomiting. So if someone takes this drug, drinks alcohol, they'll get a very nasty side effect such as vomiting. Another example of competitive inhibition is malonic acid, and this inhibits an enzyme called succinate dehydrogenase, which you find in the Krebs cycle. And we'll be discussing the Krebs cycle when we get to respiration. So here you have a graph representing how the rate of reaction changes with substrate concentration. Now notice how when you increase the substrate concentration, the rate of reaction usually increases until it plateaus at a Vmax, the maximum rate. If you add a competitive inhibitor, you'll notice that the rate of reaction will be slower. So notice that the curve goes to the right. This is because the competitive inhibitor is competing for the active site. Also notice that the reaction will still reach the same maximum rate as when there's no inhibitor. This is because the substrates will eventually come in contact with an active site. So keep this graph in mind when we discuss the next inhibitor. Now we're going to look at non-competitive inhibition, which is still reversible. So you remember that non-competitive inhibition involves something called an allosteric site, which is another site on the enzyme. So allosteric means another place. This inhibitor binds to another site called an allosteric site. It doesn't bind directly to the active site. The inhibitor forms an enzyme inhibitor complex by combining with the enzyme at the allosteric site. This alters the globular structure of the enzyme, and so you have the active site changing. So what does this mean? It means that the substrate can't bind because the active site has been changed, and you won't get a product being formed. Here you can see the non-competitive inhibitor binding in A. The active site is changed so that the green substrate cannot bind. So if you were to draw a graph of the rate versus substrate concentration, and when you add a non-competitive inhibitor, this is what you'll get. Here you can see the effect of a non-competitive inhibitor on the rate of reaction. The substrate and inhibitor are not competing for the same site. So although you're increasing the substrate concentration, it doesn't affect the non-competitive inhibitor. Now, why do you have a lower maximum rate of reaction? This is because the enzyme's not working efficiently in the presence of the inhibitor. And here's quite a nice diagram to show you the difference between competitive and non-competitive inhibition. On the left, you've got non-competitive inhibition. So this means that the inhibitor is binding to another site called an allosteric site. And it changes the shape of the enzyme so that the substrate cannot bind to the active site. In competitive inhibition, the inhibitor is competing with the substrate for the active site, and it interferes with the active site so that the substrate cannot bind. Now we're going to move on to irreversible inhibition. So what is irreversible inhibition? Well, let's have a look at this picture here. You've got your enzyme, and when your little irreversible inhibitor comes along, it binds to the active site and it binds permanently there. So the substrate can't bind. So we say that substances that can combine covalently with the enzymes and inactivate them irreversibly are called irreversible inhibitors. They react with some kind of functional group in the active site and they block the active site. Irreversible enzyme inhibitors are toxic. So what are some examples? Well, many enzymes contain a group called sulfide sulfhydryl, which is SH groups. They've also got an alcohol group, OH groups, or an acidic group on their active site. Any chemical which can react with these groups causes an irreversible inhibition. So examples would be silver, mercury, and lead. Silver, mercury, lead, if they bind to a group such as an alcohol group or SH group on the enzyme, then they'll bind irreversibly. Another example is a nerve gas called DFP. And what it does is it inhibits the active site of acetylcholine esterase by reacting with the hydroxyl group of serine. So you have this nerve gas that binds to an enzyme called acetylcholine esterase, and this is involved in nerve transmission. So in summary, what have we learned? We've discussed reversible inhibitors, and we've looked at competitive and non-competitive inhibition. Remember that competitive inhibitors compete for the active site, and the example that we looked at was disulfiram and how it is used in alcoholics. Then non-competitive inhibition, it binds to another site called an allosteric site. And then you've got irreversible inhibition. And examples are heavy metals. And that concludes our lesson. The end.